So this is a good diagram to start at and kind of review the immune cells. And in this course, of course, we're not looking at them under the microscope. You don't need to be able to identify them like that. But it's how some of them have gotten their particular names is when we were identifying under the microscope. If you take AMP2 lab, it's one of the things that you'll need to do. It's one of my favorite things to do is to look at white blood cells under the microscope. They're really pretty, but when we talk about their functionality, which is what we're really focusing on in this course, they're quite amazing in what they can do, right? So down here we have a mast cell, a macrophage, and a dendritic cell. And why do we have these guys down at the bottom of this diagram, unlike the rest that are listed up here? Where in the body would you find these cells? In your tissue, in your brain, in the tissue. As opposed to these guys are circulating in your bloodstream. Not to say you're not going to find these in other places. And as we'll start looking at, right, T cells and B cells will travel into the tissues. They're in high concentrations in your lymph nodes, which is one of the areas where your body comes in contact with antigen. The dendritic cells can travel. Macrophages can travel. And dendritic cells and macrophages are derived from what you would see in your blood known as a monocyte. Same functionality, right? They just get a little bit different names. They mature, act a little differently in different parts of the body. What's a major mediator released by mast cells, especially when we have tissue injury? Histamine, right? So this is one of the ones that's going to help that inflammation process by that histamine affecting the blood vessels. Now our lymphocytes, under the microscope, you can't tell the difference, but there's 3 different lineages that we're going to go into more detail when we get to adaptive immune response. All right, so for right now, we're like, okay, we're really going to save their functionality till we get there. Who's the major player out of these granulocytes? All these guys that are filled with granules that have toxic substances. Neutrophils. And remember, they'll be found in our tissues, but only when? When we have damage, when we have inflammation, when they're being called there. They're not normally going to go there. Same thing with basophils and eosinophils. And the reason why these 3 guys are different is because of their different granules, right? And what's inside those granules, what's inside those vesicles, give them different abilities. Which ones of these, out of all of these cells that we talk about, have the ability to phagocytose? First of all, what is that? What's phagocytose? Eating, right? They're going to engulf it, they're going to take it inside, and they're going to digest it. So who's our eaters here? Who's our phagocytic cells? Macrophage, neutrophils, one more. Related to the monocyte. Dendritic cells, right? These are your major eaters. Macrophages are big eaters, right? Macrophage, big eaters are your phagocytic cells. Your dendritic cells can phagocyte too. But their job is more important at carrying this information elsewhere. But in order to carry it, it's first got to take the information in. And it does that by phagocytosis and digesting it, breaking it down. Neutrophils can phagocytose, but also they can release these granules, right? These are really powerful killers. Again, one of the main reasons why we really only want them going into tissue when they're called there, because they'll make a big mess. Right? My nickname for them is the suicide bombers, right? They're really messy. They'll kill themselves in the process of trying to kill invaders. So how do cells, right, like these guys, detect and know what's going on in the environment? chemical signals, right? And they have receptors on their surfaces. So we saw this, and those of you guys that may not have come back after one of our tests, right? There's a list of videos for Unit 3, and we watched a bunch of them. We watched a neutrophil chasing down a bacteria. We watched inflammation, and I highly recommend, watching those if you have not, and if you did, probably watching them again. It really helps, right, when you see this stuff in action. So they have surface receptors. So this is how communication happens for our cells. They have receptors on their surface, as well as they have internal receptors. Cytokines are those chemical messages, like the hormones of our endocrine system. They even have adhesion molecules, and as the name implies, this allows them to grab on. So we really saw this in action as the cells were moving from the blood vessels, the capillaries or venules, and heading into the areas of the tissue where there's inflammation. So those surface receptors are almost sometimes referred to as the eyes and the ears, right? Allows them to see and hear their environment. The voices, these messages that we send, are the cytokines, these chemical messages released by cells that are going to be heard or picked up by other cells. Adhesion molecules, as the name adhesion implies, allows them to grab onto things, right? So very similar to our hands and grabbing onto things. So they got to be able to respond. They got to be able to do something based on what they're being communicated to them by other cells or the environment. So surface receptors are those proteins, right? When we looked at membranes of eukaryotic or prokaryotic cells, they're embedded with tons of different proteins. These act as relays to send signals inward to the cell. These receptors are specific as to what they bind to. So the molecule that actually, in general terms, that binds to a specific receptor is referred to as a ligand. So if I pass you a note in class, right, usually I've got someone's name on it that I want you to pass it to. Once it gets to that person, hopefully they're the only ones who opens up that note, right, because it's meant for them. So the same thing here. When that molecule, that ligand, binds to the receptor, right, to the cell it's supposed to be communicating with, that cell's going to respond based on that message in a particular way. Just as the person probably receiving the note, right, will respond to it in some particular way, depending on what the note says. So we can send different messages, and so can our cells. And when we look at cytokines, there are similar aspects to certain groups of cytokines. So of course we've divided them up. Scientists like to categorize things into several different classes. And these are some of the more predominant classes of interactions that we see. So a whole group of cytokines are referred to as chemokines, referring that they're important in chemotaxis, which is movement in response to a chemical signal. And we saw this, right, when we talked about bacteria moving, right? They have receptors on their surface. They can respond. If they have flagellar, they're going to go towards something if they like it, and they're going to head away from it if it's bad news. Where do all your white blood cells come from? Even your red blood cells. Where are they made in your body? In your bone marrow. How does the stem cells in your bone marrow know which set of cells we need at any one time? They got to have some type of communication, right? The body's got to say, hey, we're using a whole lot of neutrophils. You better make a whole bunch more. All right? We got a serious problem afoot. So they're going to send that signal. Because that's a particular group of cells, or a colony of cells, a grouping of cells, is referred to as a colony stimulating factors. So there are specific ones that will tell the specific stem cells what set of cells to make. What set of white blood cells or leukocytes are needed at any one time. All right, we're going to communicate this information chemically. Interferons are a set, and of course under a lot of investigative research, because they interfere with viral infections. And so we'll look specifically at that interaction later and how interferon works. And so they've even used this as an antiviral treatment, where they give this cytokine in excess to people to interfere with virus infections. Interleukins, inter means in between, and lots of different white blood cells communicate with each other in order to mount an immunological response. So they have to be able to communicate with each other. And this is particularly important when we go from innate, our first line of defense, to our adaptive. We've got to be able to communicate the information from one group of cells to another. So a lot of times, and I've even heard it in some of the videos and stuff that I've been watching, that you guys have been posting, IL-1. It actually stands for interleukin number 1. So it was one of the first ones they discovered, of course, so it got the number 1. And it can communicate, it can be released by certain cells to communicate with other cells a particular signal, right? What's going on? What do we need to do next? So there's tons of interleukins, and there's probably still discovering more each day, and discovering more about which cells release and which cells respond in what particular way under what particular circumstances. The good news for you guys is we're not going to memorize any specific cytokines, just the generalization. Tumor necrosis factor, as the name implies, these have the ability to kill tumor cells. So, of course, another active area of research. How is this working, right? How is this effective? How could we improve upon what the immune system is already doing to help fight disease? So, as I said, here in this table, they actually list some specific interleukins, right? So you have IL-1, as I said. Macrophages and epithelial cells can release this. If they bind to a T cell, that could activate it. If it binds to a macrophage, it could activate that cell. It can also induce fever. So there are a lot of interleukins. I forget what number they're up to now. I haven't kept up with the latest research. When I was in grad school, I knew up to number 20. Of course, I don't use that information anymore, so I couldn't tell you what IL-20 is released by and communicates with. But you can look it up in a book, right, or on the internet. We don't need to memorize lots of data that we're not using on a regular basis. But it's almost like a foreign language for some people, right? All these acronyms and such that we use. So the good news for you guys is we'll keep it to a minimum, right, as far as the acronyms and such go, and how many things we're memorizing. The important thing is understanding that there's communication here, right? And it's specific communication between specific cells. Adhesion molecules are pretty straightforward, right? They allow the cells to literally adhere to each other. So we saw this in that animation, and we witnessed the result of it in the videos that we watched, where you literally saw the white blood cells adhering to the endothelial layer of the blood vessels because they're grabbing on to those receptors that are being expressed so they can move to the area where they're being called. Well, how do we know, right, the bad guys have gotten in? We can sense them. We have a general set of danger warning receptors on our cells and on our white blood cells. We also have a system of proteins that can be activated that are found in our blood and our tissue fluids. We can even detect double-stranded RNA, which, do we have double-stranded RNA? Who would have double-stranded RNA? What type of pathogen? Viruses, right? Remember, viruses can have DNA or RNA. It can be single-stranded or double-stranded. So right there's a dead giveaway, right? That's something foreign, right? Double-stranded RNA, not common to our cells. So we have systems within the blood that detect signs of tissue damage or microbial invasion. They're responding to patterns, right? So these common molecular patterns, right, that are associated with microbes. Some of these can directly, when they detect, destroy the invading microbe, or they're going to call others to the area, or they'll do both, right? They'll kill it, and they'll call others to the area to kill others that they are not able to eliminate. So what's some molecular aspect we know about bacteria? What are some of their molecular makeup? Something that would be what a molecule is associated with just bacteria. Plasmids are circular pieces of DNA, very common to bacteria, but think outermost covering. Capsule, then that one's tricky, right? For some reason our body isn't very good at detecting that, but cell walls, we know that there's a unique substance for bacterial cell walls. What's that? Peptidoglycan. So if we have a receptor in our body that can sense the presence of peptidoglycan, we know that's not us, right? We know that's bad guy, potentially foreign, damaging. And again, it's unique to bacteria. So do you think we have receptors that can detect peptidoglycan? You betcha, right? Because right away that's going to send the alarm that, you know, we have an invader, a bacterial invader at that. What about gram-negatives? What's their outermost covering? It's an outer membrane. Does anybody know a substance that's found in their outer membrane? LPS, lipopolysaccharides. Teichoic acid associated with gram-positives. Right? These are generalizations, right? There's lots of bacteria that are gram-positive, lots of bacteria that are gram-negative, right? That have these molecular attributes. But for the most part, for us, bacteria getting inside of our body. Now remember, your intestines, although it seems like it's inside, is an external environment. We're talking about something get into your tissues, right? Deep into your skin. Your cells are going to not want to see peptidoglycan there, or lipopolysaccharide, or teichoic acid. That's going to be a danger signal. Yeah, so our normal flora, even when it breaches our barriers, when it gets to where it's not supposed to be, we're going to respond to it. Because again, it's foreign. It's not where it's supposed to be. So our normal flora, remember, in our intestines, that's really not truly inside your body. It's truly outside. Right? It's only when it gets out of the intestines into the body that we're going to respond to it. Okay? On the top of your skin, again, it's not in the deep layers of your skin. So we're not going to respond to it. That's why sometimes your normal flora can be a source of infection, because they get to other parts of the body that they're not supposed to be in. Does that make sense? Excellent question. Thank you, Tyrone. So again, the very first thing is we have these barriers to help protect entry, but stuff is going to get in, right? And when it gets in, we need an alarm to go off to let us know that it got in, and we also need to try and destroy it, right? We're not going to just go, oh, you know, someone else can deal with it. No, right? Some of these guys, their job is to actually kill it. So very simply, right, we're looking at patterns. Again, microbiologists, for some reason, immunologists like to use a lot of acronyms. I won't use these particular acronyms, but they're out there, so that's why I have them listed on here. The important thing is that these are receptors that recognize specific molecular patterns that we associate with danger with microbes, right? So things like lipopolysaccharide associated with the outer membrane of gram-negatives, peptidoglycan, right? Definitely the outermost covering maybe of a gram-positive, lipotectonic acid associated with gram-positives, lipoproteins, again associated with some bacteria, flagellin subunits, this is the protein that makes up the flagella of bacteria, and then especially viral RNAs. Right? We can detect that different RNA from our own, especially when it's double-stranded. So, you know, they're common molecular components. So Toll-like receptors are a specific group, right? So this is a specific group that we've studied, that they know what the receptor is binding to, what signal it's sending. So I'm going to mute the audio on this one. And so, of course, these are proteins embedded what? In our cells' membranes, right? And they're receptors for specific substances, right? So I'm going to let it. And so say this is a phagocytic cell. Well, one of the great things about being a phagocytic cell is that if it detects it, it's also going to what? Engulf it, eat it, and destroy it. Right? So it's not only going to just maybe send out a signal, it's going to say, here's the bad guy. So I'm going to fast forward this because I want to get to a particular spot and pause it. Okay. So Toll-like receptor number 1, number 2, right? Number 3, 4, whoa, wait a minute. 3. Where is that? It's inside the cell. And if you look on the list over here, what does it have the ability to detect? Double-stranded viral RNA. Are we going to be able to detect that outside of our cells? No, it's not until it gets inside of our cells, gets out of that protein coat, that capsid, that we'd actually be able to detect it. So having that receptor on our surface would do us no good, right? We need it inside the cell so that when they engulf it, right, they can recognize it. But other things like cell wall components or flagella, right, notice these receptors on the outside of the cell. When these things bind, and this is where, you know, you watch the video to fall asleep if you're getting tired and you can't fall asleep. Because they'll get to the really complicated components. So here it's binding with the receptor, right? That particular component. In this case, it's lipopolysaccharide. These are receptors. They're carrying a message. So there are other cells on the other molecules, on the inside of the cell. They're going to carry this message, this signal, to other parts of the cell. And they go into specifics and give all these guys names, I think. Yeah. But the important thing is where did that message go? You see where it just went? It went where? Into the nucleus, right to the DNA. Why'd it go there? DNA is what? Information. The nucleus is the information command center. If a cell needs to respond, it's going to need to check with its information bank and respond accordingly depending on what that signal is, right? So if it is to release a cytokine to warn other cells, where is that information stored? In the DNA, right? So it's going to interact with the DNA and say, okay, make interleukin number 2, right? And then this cell is going to secrete it to warn maybe other cells of the invasion. It's not going to just take the message and do nothing. It's going to do something. And how it's going to do that is going to be determined on the information signal the cell received. Right? And this information goes right to the nucleus. So what's it going to cause? Things like gene expression, right? You got to use that information that's in the DNA to do something. The most common responses that you're going to get is inflammatory response, where they're going to call other cells to the area. Right? If we have an invader, we want others to come help eliminate it or detect it. And then we respond in a particular way when it comes to viruses. And that's a little bit different than inflammation. So again, notice receptors on the outside of the cell, but also receptors on the inside. So notice even like if a bacteria gets on the inside, we can detect bacterial DNA versus our DNA. Any one of those receptors that triggers a message is going to be sent to the nucleus, and the cell is going to respond. Right? It's going to activate some type of information, some genetic information, and it's going to do something. The most common overall responses that we see are inflammation and then specific antiviral responses. So who have we found Toll-like receptors on that can recognize these repetitive patterns that we see with things like microbes? Well, of course, the macrophages and the dendritic cells. Because remember, they're right there at the tissue level. They're our scouts. The macrophages are really going to be the major eaters, but they can also send signals to other cells. And the dendritic cells are going to eat, but their major job is going to be to send this information to our adaptive immune response. Right? And so notice cells lining sterile body sites. Right? Where stuff is not supposed to be. Right? When it breaches into the deep skin, it's not supposed to be there. In our muscles. Right? They're going to be like, oh, no. You're not supposed to be here. I don't care if you're normal flora. You're not where you're supposed to be. Therefore, you could be a bad guy. So NOD-like receptors are, again, just a group of receptors. The difference here, these are ones in the cytoplasm even. So again, this multi-layer of detection. We don't only can detect on the surface of those cells, but inside of our cells. And the most common response, of course, is going to be inflammation. If our cells detect danger, right? Molecules getting in that aren't normal. So, for instance, one of the ones they have here is uncapped single-stranded RNA. When we are making our RNA, we cap it, right? We molecularly modify it. And so if a cell runs across single-stranded RNA that's not modified, it's not capped, how we usually modify our RNA, that's a danger signal, right? This is probably foreign RNA. And we need to respond accordingly. Even have ones for like when our cells themselves get damaged, right? Be able to recognize that they've been damaged and send the signal to other cells. And again, notice that some of these events will lead to the destruction even of that cell. But we're thankfully not unicellular organisms, right? We can lose some cells. And trying to keep the whole organism, right, the host in the case of when we're having invasion, alive. We have cells to spare. So I'm not going to play this animation, but you'll notice that some of my animations I have embedded into my PowerPoint, but I also have them all on that list with web links on that page. I'm just going to fast forward to a particular spot that I want to show you guys. I guess I'll probably start right here. So this is very similar to what we saw before, right? We've got some type of injury. Even worse, we've got some type of invasion, in this case a bacteria. So cells in that area, right, are being damaged. Receptors may be triggered, and they're sending out signals. As far as the complement system goes, this is in our tissue fluids already. And this is a series of proteins that we've labeled with the letter C, and we've given numbers to them and additional letters because some of them break into pieces. So you have C3A was once C3, right, that broke into two pieces, C3A and soon to see C3B. There's B. So these proteins have abilities. So C3b actually binds to the surface of the bacterial cells. It doesn't bind to our cells. Our cells have regulatory proteins that keep it from binding to our cells. What happens when C3 binds, it's like tagging them for destruction. Phagocytic cells like this neutrophil have receptors on their surface for C3b. So when they come along, they see C3b attached to a bacteria, they're going to gobble them up and destroy them. They know it's tagged, they know it's a bad guy, they know it needs to be destroyed. This attaching a molecule that causes phagocytosis to happen is referred to as opsonization. So other molecules that you see are C5a. So again, this is another component that broke into two pieces. These protein components, one of them in this case is tagging them. Others in this case are very important for communication. So in this case, C5a is specifically calling, it's a chemoattractant, specifically calling the neutrophils to this particular area. In addition, if you roll back, notice C3a, its effect is on the capillaries itself, allowing them to become leaky and dilated so that the phagocytic cells can come out of the blood vessel and head to the area where the C5a is calling it there. Gobby, gobble, gobble. The next thing that can happen is that the C5b and several other ones, you guys don't need to memorize them, form a particular complex we refer to as the membrane attack complex. This can form this donut-shaped protein matrix in membranes. When it does that, it essentially pokes a hole in the membrane and is going to cause lysis of that cell. So this can lead to direct destruction of cells. So these are the major outcomes, and I'll go back to the first slide. Right? So these are the really important proteins in this complement cascade. These are the important outcomes of things that can happen, right? Inflammation with the use of C3a and C5a. Opsonization, right? That tagging for destruction by C3b. And then cell lysis by several of the proteins all together we call the MAC complex, right? Membrane attack complex. So as I said, this is the major important thing, right? Inflammation, opsonization, that tagging for destruction by phagocytic cells, and lysis of foreign cells. The proteins, as I said, are given the letter C to denote the complement system. They're numbered 1 through 9. Unfortunately, not in the order in which they're truly activated, though it kind of goes. But in the order they were discovered. Some of them break into two pieces, two fragments, to indicate that it's a fragment, right? We have the A and B designation. So constantly C3 is split into C3A and C3B. And when the C3b binds to bacterial cells or foreign cells, it keeps this reaction going, right? It's almost like a cascade, almost like a domino effect. We refer to that as the alternative pathway. And this is the other bad news about that. It sounds like that's not what usually happens, right? It's an alternative. No. That's probably what always happens first. Because the classical pathway, which we'll learn about, is initiated by antibodies, which are a product of your adaptive immune system, and they take time to be produced. So unless you've been vaccinated or you've had prior exposure, complement's probably not being activated by antibodies. Instead, it's C3b binding that's activating it and keeping this cascade of events going. Right? So a few letters and numbers that you do need to memorize because they're important are the C3a and C5a. So as we saw, these guys are really important to inflammation, making those blood vessels leaky, getting those neutrophils to the area. And then C3b, as we even saw before when we looked at the details of phagocytosis, phagocytic cells like neutrophils and macrophages and dendritic cells have receptors specifically for C3b. They'll grab onto it and engulf and destroy those cells. When you put a tag on, like C3b, and it causes phagocytosis, this is referred to as an opsonization, right, or an opsonin. Anyone know the other powerful opsonin that our body produces? That if we coat a bacteria in it, phagocytic cells will engulf and destroy it. It activates the classical pathway of complement. It's a product of our adaptive immune response, and it's a protein as well. Good old antibodies. The difference there is antibodies are highly specific to what they're binding to. C3b, not so much. Right? This is a general protection. So that cell lysis, right, C5a, C6, 7, 8, and several 9s. Don't need to memorize that list. Right? Just know that we call this the MAC complex, the membrane, and that's the important part there. This only inserts into membranes and creates the pores. So, it's most effective against gram-negative cells, less effective against gram-positive. Why? What's the difference between these guys? Which one has two membranes? Gram-negative, and actually one of those membranes is the outermost covering. We call it the outer membrane. So these proteins can very easily insert into that outer membrane, where for a gram-positive cell, those proteins would have to travel through the cell wall to get to the membrane, the one membrane they have. It can be done, just not as easily as with gram-negatives. I wasn't kidding when I told you guys I'd never shut up about gram-negative and gram-positive. All right, so there's an electron micrograph showing, look at all those dark spots. Those are all the holes, right, from MAC complexes inserting into a cell and destroying it. They ain't messing around. So, as I said, it can be activated, I've only mentioned 2 ways. In truth, it can be activated 3 ways. In this animation, unfortunately, I only show you 2. I don't want it talking in my ears. I just want to. So again, the important things that we've already talked about, right? What's going to happen when these guys get activated? We're going to get inflammation. We may even get cell lysis, right? We may even get opsonization. So this, as I said, is the classical pathway. So as I said, you've got the antibodies binding to the cell. And this is a specific interaction. And then complement proteins can interact with antibodies that are bound to bacterial cells like seen here. And it's going to start this cascading event. And the good news for you guys is not a graduate level immunology class, so you don't have to memorize all the steps. We're just going to start at the beginning and jump to the end. right? So it gets activated. What are you going to get? You're going to get the usual outcomes. Now, for the alternative pathway, it's all about C3, becoming C3A and C3B. And that's because C3B bound to the surface of the cells, right? It's not being regenerated. So most of the pathways, once you get to C3 being converted and being split continually into A and B fragments, the pathways are very similar, right? So as we saw, when you have lots of C3A, you're going to get leaky blood vessels, you're going to get inflammation. C3Bs are binding, they're opsonizing to bacterial cell walls if they can. And then that's going to cause them to get gobbled up and eaten by phagocytic cells. And the next step, the major step, is C5. And so again, C5 is really going to call those neutrophils to the area, and then C5b starts that formation of the MAC complex, that membrane attack, where we go poop, 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 poke a hole. So series of circulating proteins, right, in blood and your bloody fluids. They're inactive. They got to be activated, right? So whether it be by antibodies or C3b binding, and we keep getting C3a and C3b, they help us with the adaptive immune response. And once we stimulate the inactives, right, whether it be antibody binding or the C3b binding, it's a rapid sequential activation, right, like a domino effect. So alternative, although as I said, we found out about it after classical, it's probably what happens most often. Lectin is a little bit more specialized. And that one is initiated by mannan-binding lichens. These are special proteins. Again, pattern recognition. So mannan is a polymer of lots of mannose sugars hooked together. These are found in microbial cells, not in our cells, this polymer of sugars. So we have specific proteins that can detect the presence of them and activate this system. So notice, C3, C5, right? Those common outcomes, inflammation, cell lysis, opsonization, can also happen. So all these guys, whether it's triggered by alternative with C3b binding, or you have the mannose lectin binding proteins activating it, or antibodies, we're going to get to the point where C3 is converted and continually broken down into the two components A and B. B can bind to cells and cause them to be phagocytosed, what we call opsonize. The C3, and then later when 5 is activated in the cascade, C5a, both of these are going to induce inflammation. And then we can also have the formation of that MAC complex. So 3 different ways we can trigger, 3 different possible outcomes that can happen. All 3 of these could potentially happen. You could have C3 binding and opsonizing and destroying. You could have cell lysis destroying. You can have inflammation. Most commonly, you should always get inflammation. You're not always going to get C3b binding, and you're not always going to get cell lysis by the MAC complex. Because guess what? Some cells can stop our processes from going forward. So remember I said our cells are protected. So here's our host cells. We have complement regulatory proteins on their surface. So C3b can't bind. And if it does, it's inactivated. So it can't convert into more breaking down of C3 into C3a and C3b. So it can't activate that process. So what happens for microbial cell walls is they don't have complement regulatory proteins. So C3b binds, right? It's able to activate the other components, and complement system becomes activated. The bad news is that some bacteria have learned to use and steal our regulatory proteins to deactivate C3b from binding and activating the complement cascade by the alternative pathway. Tricky little guys. It's like walking into the hospital, putting a lab coat on, and walking right through into the operating room, right? They think you're a doctor. You put your complement regulatory proteins on your surface, our system's going to be like, well, you're supposed to be here, right? No, they're hiding. All right. Yeah, we'll go through this last one, because I think this is our last one for, oh, and then fever. We can hack it, right? Okay, so cell gets infected by a virus, right? Virus gets inside of our cells. It's going to become unpackaged. It's going to start turning our cell into virus-making machinery. But remember, we have receptors inside these cells. The cell detects, oh no, right? There's viral RNA present. One of the common responses it can do is release a cytokine we know as interferon. Interferon binds to neighboring cells, basically is a warning signal that says there's viruses abound. Prepare yourself for war. So what that cell that has interferon binded to it's going to do is it's going to produce digestive enzymes that will destroy messenger RNA. So if the virus gets in, it will destroy that virus's RNA. The bad news with this is that we can't really regulate what messenger RNAs it destroys, and so it kills the cell itself in the process. But what has it stopped that we really need to stop? Replication and therefore spread of the virus. Right? So again, we're multicellular organisms. We can afford to lose some cells, right? And especially the guy who sent out the warning signal, right? The one that released the interferon to begin with, he's definitely toast. And even worse, he didn't prepare ahead of time, right? So he becomes the source of the virus. It leaks to neighboring cells, but the good news is he was able to scream out, right? Send that interferon, tell them, get prepared. Bad guys are nearby, just in case it comes your way. Defend. And they're going to defend by even just destroying themselves, basically, in an effort of stopping that virus. So the interferon causes the neighboring cells to express inactive antiviral proteins. When the virus gets in, though, it's going to be activated. It'll stop protein synthesis and it'll cause that cell to go through apoptosis, which is programmed cell death. Right? That cell is going to die. So remember I said, right, we can detect their RNA is different from our RNA. It often has 3 phosphates at the 5' end. Unlike ours that I said, we cap it, right? We put phosphates on it. We tag it in a particular way. And again, our RNA is never double-stranded, but sometimes RNA from viruses can be double-stranded. And that's a big danger signal to our cells. Like, no, this is not right. Right? This molecule is not supposed to look like this. Fever. Is fever a good thing? It can be, right? Because what do we know about microbial growth if you get too hot? you will kill them, right? What molecules in their cells are you disrupting? Well, no, mesophiles are the ones that if we get too hot, we're going to affect, right? Because they're the ones that live in us. But molecularly, what are we damaging for them that they really need to function? What are the workhorse molecules in your cells? Proteins. We can also disrupt membranes with too much heat. Problem with this is, what happens to our cells if we get too hot? We start disrupting our own proteins, right, in our own membranes. And this is why if fever goes too high, it can be bad, right? That's why sometimes when we think fever, we're like, oh no, right? We get nervous. But this is, for the most part, a normal immunological response to infection, because it can slow down or kill microbes, and molecular processes themselves happen faster the warmer you get, right? Molecules move faster in warmer environments. Fever itself is one of the strongest indicators usually of bacterial infection. Because although, as I said, our body will normally increase its core body temperature under certain infection situations, unfortunately, some microbes, like bacteria, produce substances that will cause us to heat up above what we would normally do. So, cryogens are fever-inducing substances. We have our own, right, endogenous internal cryogens that'll cause our core body temperature to increase. But microbes can excrete, right, exogens that also can induce fever. So this is where you have a problem. When fever is long-term, not going away, and increasing, it's usually a pretty strong indicator of a viral infection. When you have intermediate fever, right, up and down, it's usually viral infection. And again, it's our body that's causing us to warm up in the case of the viral infection. In case of bacterial infection, it could be our body and the bacteria. So that combination is a bad combination. Now, why on earth, why they would make us so hot that they would actually kill us? Doesn't sound like a good evolutionary pick, right? Kill your host. But some of them, will just keep eating us even after we're dead. So some of them don't need us alive to actually eat us. So making us too hot and killing us off wouldn't necessarily be a bad thing for them. And obviously they're resistant to that high temperature if they're releasing substances that would have us increase in temperature. But what are the benefits, right? The benefit for us to do it, to increase core body temperature, is we're going to be able to grow faster, right? Produce cells faster, metabolic processes faster. And hopefully we might exceed the maximum threshold for a microbe like a bacteria and actually kill them off. Make sense? Okay. So how many people thought fever was just bad? Yeah. So there are good aspects to it. There is a reason, and we do actually do it naturally, right? But it can be induced by microbes as well, and that's the bad thing. Again, using our own system against us.